脑人言。
是来这边跟我们大家分享最近的他的研究成果。这两篇文章是分别于去年9月份和今年1月份在那个 Neureport 接收的。它是专注在研究纹状体通路在抉择任务当中的功能。那我们首先来介绍一下今天的演讲者唐顺航博士。他之前本科毕业于中国农业大学，之后到中国科学院神经科学进行博士的 training， 在徐宁农实验室。之后在徐宁农实验室继续进行了博士后的研究，目前也正在准备要在找国外的博后。所以如果我们的听众里面大家有推荐的实验室或者有感兴趣的，可以跟我们作者联系。那今天要介绍的内容主要是纹状体的直接和间接通路在听觉介导的抉择任务当中的功能。两篇文章，是唐顺航博士和崔乐乐博士共同合作完成的。他们分别主要专注在他们神经环路的调控以及成像的机制方面。接下来就由唐顺航博士来给大家详细的介绍一下这个非常有意思的工作。
好的，感谢文平师姐的介绍。稍等一下，我分享一下屏幕。
好的。
从这头开始。OK， 可以看到对吧？
现在可以看见了，刚刚
现在可以。
好的，
OK， 好的。很高兴今天能在这里跟大家分享我们最近发表的工作。然后我后面的就是 talk 可能是用英文讲的。然后但是大家如果有问题的话，反正用中文或者英文提问都可以。So let's start. So hi everyone, I'm Sheng Hang, and thanks for the invitation on your report. It's my pleasure to be here, share our recent work. So in the next 1 hour, I'm going to tell you whether and how does the striatal circuits play distinctive roles in perceptual decision making. I hope you will enjoy it. So what is perceptual decision making? Imagine when you walk on streets and you saw a mushroom in your way. So it gives you some sensory input, and it's better to make a choice to jump and to avoid the clearing. And so in this process, you're making decision based on the sensory input and commit the choice with select the action. So as you can see, perceptual decision making is a basic but very important cognitive function. So how does the brain make perceptual decisions? There are many brain areas considered to involve in this process, and the basal ganglia circuitry for a long time has been considered to involve in the many cognitive functions, including action selection and motor control. So the striatum is a major input nucleus of the basal ganglia, and it receives abundant input from cortex, thalamus, and midbrain dopamine neurons, and also it sends up to the downstream nuclei of the basal ganglia nuclei. So previous study have revealed that striatum is involved in perceptual decision making. For example, in this study, they trained the monkey to do the classical random dot motion discrimination task, where the monkey is required to report the direction, motion correct direction of the moving dots by secant to the target. During this task, the micro stimulation of the striatum would bias monkey's choice towards the other direction. But simply knowing striatum is involved in this perceptual decision process is far from enough. Why? Because unlike the cortical area, there are two major types of the projection neuron in the striatum. The direct pathway spinning projection neuron, and we call it DSPN, and another is indirect pathway striatal projection neuron. It is ISPN. So these two types of neuron give rise to the famous to direct pathway and indirect pathway of the basal ganglia. So as their name, the DSPN had direct input to these basal ganglia upper nuclei, the GPI and SNR. But for indirect pathway, it can only indirectly influence these nuclei through their projection in the GPE. And for these two types of neurons, they also differ in their like the molecular markers. For DSPN, they express D1 type dopamine receptor, and ISPN, they express D2 type dopamine receptor, and also the A2A receptor. So it is important to distinguish the function of these two types of neuron in the perceptual decision making. And traditionally, these two types of pathway, also these two types of neuron is considered to be involved in motor control. And the classical view think they have opposite role in motor control, which is the direct pathway promotes a movement, and the indirect pathway would suppress movement. So this view is supported by plenty of experimental evidence. And for example, this optogenetic experiment, when they optogenetically activate DSPN, the mice locomotion increase. And when they activate the indirect pathway neuron, mice locomotion was decreased. And also for this model, they further predict that during voluntary movement, the direct pathway and the indirect pathway would display such opposite activity change. And however, so recent reported calcium recording result was against this prediction. They used fiber optometry to record and found that both DSPNs and ISPN, their activity increased when mice turned to the contralateral side. So so as this schematic shows, also several other studies how this co-activation pattern of these two types of neuron, and some paper show that the DSPN and ISPN have indistinguishable activity pattern. So this would raise a contradiction that how would this indistinguishable activity pattern with the opposing pathway would generate selected action. And people also raise another alternative hypothesis, which is concurrent but differentiation activation of the direct pathway and indirect pathway. But still, this hypothesis lacks direct evidence support. What we want to ask is, how about their function during perceptual decision making? Can we have a clear answer compared with this the mass of the study. To study perceptual decision making, we use the auditory guided perceptual decision, which we previously developed in our lab. We trained the head fixed mice to do an auditory guided perceptual decision making task, where the mice need to report their choice by licking left or licking right to report they are having for example, a low frequency tone or a high frequency tone, and they would give what a reward if they have the correct choice. And from this plot, the gray bar indicates the sound, and this red dots and blue dots are their mice licking. So you can tell after the sound, the mice will quickly respond to the sound tone, and because we deliver a series of tone frequencies, and if we plot their choice fraction under these different tone frequencies, we got this S-shaped psychometric curve. And this S-shaped curve indicates that the mice was really doing perceptual decision. So under this well-controlled and highly repeatable task, it would help us to find out the more delicate function of DSPNs and ISPNs. The brain area we focus on is the posterior dorsal stratum. Here, for this part, why we choose this part, and this part receives really abundant input from like the auditory cortex and the auditory thalamus. So since our task is an auditory guided perceptual decision, we think and there's also some evidence to show the projection from the auditory cortex to this area. They have been involved in the learning of this perceptual decision. So next, we ask in our task, does the PDS, the posterior dorsal stratum, is involved? So we use the inhibitory opsin RHT to test this. We express the RHT in posterior dorsal stratum and unilateral deliver the light to inactivate this area. So we plot the photo simulation trial and control trial with which is without photo simulation separately. As you can see from this plot, when the compare with the control trial, the photo simulation trial, the mice choice was biased to the ipsilateral side, which means the contralateral choice is impaired. And so sorry to forget to mention that. So the controversial choice in here means the opposite side towards the simulation hemisphere. And we also did a GFP control. We express the GFP in this area and also do the photo simulation. There's no behavioral effect. And also, we also chose another more anterior dorsal stratum subregion as to test whether this PDS is specifically involved in this task. And surprisingly, when we inactivate this more anterior dorsal stratum subregion, there's also no behavior effect. So, which means for this subregion of the stratum, it is regional specific for this task. And as we previously mentioned, so knowing only the stratum is involved is enough. So next we test the cell type specific causal involvement. So here we use pre-mice to label the DSPN and ISPNs. The D1 pre-mice is used to label DSPN, and D2 pre-mice for the ISPN. And so when we express the excitatory opsin ZHR2 in a query-dependent manner. So when we use photostimulation to activate the DSPN in the unilateral dorsal stratum, so here the blue curve is photostimulation trial, and the gray curve is control. Compare with the control trial for when we activate unilateral activate DSPN, the mice choice was biased to the contralateral side, and when we unilateral activate the ISPN, the mice choice was biased towards the ipsilateral side. And here the photostimulation length, the duration is covered the 1 second after the sound delivery. And so here we show a leaking pattern and also the answer time of the mass choice. As you can see from the 1 second from the sound delivery, most of the reaction, most of the answer was already happened. And here the red dots indicates their contralateral answer time, and the blue one is the ipsilateral answer time. So if we further divide the period into two periods, first is the first half of the sound delivery period, and second is the second period of the sound delivery time. So we can see that during the end of the first period, there are a lot of the answer leaks. So from there, we can hypothesize that during this first period, there would be the choice formation time, and after that, most of the trials they already formed the choice, and they are doing the choice execution or the water consumption. So next we use we take advantage of the genetics to do the further time restricted activation. For this we did for the ISPN, and for the first period one, when we photo activate the ISPN, they show this impairment of the contralateral choice, but not for the period two. If we compare this two period with the whole length, the 1 second optogenetic photosimulation, we can say that even we restricted the photosimulation within the very first very short period, there's also a significant impairment of the contralateral choice. So from this, it suggests that the involvement of the ISPN is more likely to during the decision formation period. And also, as you may know, that the problem with optogenetic activation is that it often raises the activity level to the more higher activity level than their psychological condition. So we also did the inactivation experiment. So for the inactivation experiment, we also use the crate-dependent manner, and we express the inhibitory opsin NPHR. So for this DSPN, and when we unilateral inactivate DSPN, the mice choice was biased towards the epilateral side, and so I'm not sure if you notice that there's there's a difference. There seems a difference for the middle tone and the end tone. It seems that the middle tone has a larger effect. So we further quantify this difference. And if you separate this tone frequency into like the middle tones and the end tones, you can see that there is indeed a larger effect for the middle tone, and also the stats confirm that. And so why that happened? What's the difference between the middle tone and the end tones? One possibility is that the middle tone is more difficult and more cognitive demanding. So there shows this effect. And also we can quantify the effects in another way. As we previously mentioned, we use the psychometric curve to fit their behavior. And here's the psychometric function, where this U is gives the categorical boundary of the curve, which tells you at which point there's equal chance for the mice to distinguish to consider the tone as equally high frequency or low frequency, and this parameter will tell you about their perceptual process. And if we plot for this example session, if we plot both the control trial and for the simulation trial, you can see that for the categorical boundary, the photosimulation trials, their categorical boundary is shifted towards the higher frequency. So for all the session, we found a consistent effect for the simulation, and this also indicates that when we inactivate the DSPN, there might be the perceptual decision formation process was influenced. And also, we can do the same thing for the ISPN. When we unilateral inactivate ISPN, there's also a small but significant effect for the choice bias. And also, it is consistent that their categorical boundary is biased. And you can notice that the bias direction for these two type of neuron, there's opposite. So for the inactivation experiment, the effect of the DSP and ISPN is also opposite. And previously we mentioned some photometry recording reveal that both DSP and ISPN is activated. And we think, is there possible a way to co-activate these two pathways? And one possibility is that there's a PV interneuron within the stratum. They do not project outside to the stratum, but they innervate both DSP and ISPN, and also this is inhibitory innervation. So first, we look at whether their PV activity reflects some task variables, and we use photometry to record from the PV neuron or the PDS, and surprisingly, we found that for the PV neuron, here you can see that the color indicates their activity level. And for PV neuron, for the contralateral trial and the ipsilateral trial, their activity level are similar. And a cross-section result also tells the same result, which means for the PV neuron, when during the mice performing the task, they do not selective to either contralateral choice or ipsilateral choice. Rather, they are activated simply by their choice. So given this, we can use the PV neuron as a tool to co-activate DSPN and ISPN. So we use a similar way to express the inhibitory opsin MPHR for the PV neuron. When we inactivate the PV neuron, because PV inhibit both DSPN and ISPN, which means the PV is inhibited, so there's the disinhibition of the DSPN and ISPN. For their choice behavior, the mice choice was biased towards the contralateral side. And as you may remember, when we unilateral activate the DSPN, there's also a contralateral side for their choice behavior. So this experiment indicates that when we co-activate DSPN and ISPN, the effect of DSPN is likely to be dominant. And so this gives us a hypothesis to test whether the activity for DSPN and ISPN also can show some trend of this difference. And we do two-photon imaging of the DSPN and ISPN during mice performing the task. We deliver the GCAM sensor 6S, GCAM 6S to either D1 crate mice or D2 crate mice. And we implant a cannula window, which allows us to directly optical access to the calcium signal of the striatum. And during mice performing the task, you can see from this example field, there are 4 example neurons, and they would activate at different time. So next, we want to quantify what's this activity relate to their task performance. So we further sort their activity into as different trial condition. Here we sort their activity according to their choice is contralateral or ipsilateral, and also their sound stimulus is low frequency or high frequency. So here each row is one trial, and also the color indicates their activity level. And we align all the trials with the sound onset time. And here there's a magenta line, as you can see, is the mice onset time. So from this plot, you can tell only these 2 panels, there's high level activity, and also the time is well aligned with the choice rather than with the sound delivery. So we can see this neuron, it prefers the contralateral choice, and also as the matrix shows. So within the same field, we also found another DSPN. It shows high preference for the ipsilateral side. So within the same imaging field, there's a certain level of DSP the response. They would both either response to the contralateral side or the ipsilateral side. And also for ISPN, we also found a similar heterogeneity within the same imaging field. And further, we want to quantify this heterogeneous response. What's their contribution for different task variables? For here, as you can see here, there's at least there's choice and sound. And so we use we use a linear regression model to do this. And so first we chose 3 types of variables. First one is the sound category, which means the sound is a high frequency tone or low frequency tone. And the second is a choice, and the choice is either contralateral or ipsilateral, and also the outcome, which means it is a rewarded or no reward trial. And also we use the kernel like function to further differentiate their timing information of the response. So if we show the every neuron we recorded from DSPN and ISPN, and here we simply so each row indicates 1 neuron, and for each column is for different task variables. The first one is sound category, also indicate here, and the second one choice, the third one is outcome. And here the blue one and also the red one, the green one indicates significance of the regression results. As you can see, for DSPN and ISPN, for the significant responsive neuron, they are mostly the majority is choice selective neuron, and if we quantify their explain variance, unique explain variance by calculating the CPD or the regression, we can tell that for the encoded information, it is also the choice is dominance. And so next we want to see what's their choice preference. And so we calculated the selective index, which is also derived from the receiver or operator curve. And so we can use this to quantify their preference for the contralateral choice or ipsilateral choice. So here for this plot, each row is 1 neuron, and the color indicates their choice preference. Red is more red means controller, and the blue indicates ipsilateral. So you can see that both DSPN and ISPN, they also have different group of neurons respond to contralateral side or ipsilateral side, and but I think the contralateral choice prefer neuron is dominant, and so further we can look at their preceding choice period, because it is likely this period would contribute to the choice process. And for this preceding choice period, there's also preferring control neuron or preferring ipsi neuron. But the fraction between DSP and ISPN, they look similar, right? And so this also raises the similar contradiction. If with this similar fraction of contra-preferring neuron and ipsi-preferring neuron for the DSPN and ISPN, and also they have this opposing role for the choice behavior. How would they coordinate to support desired action? It's likely that you cannot decide either action. So next we look into more detailed activity, and here we plot the activity from contra-preferring DSPN, and we for also each row is one neuron, and you can see that as expected for the contra-preferring neuron, the contra choice activity is higher than the ipsilateral choice, and also this is the main trace. And but if we further calculate their choice preference by subtracting these two curves, and so you can see that before the answer execution, this contralateral preference was rise higher. And also we can do the same thing for the contralateral preferring ISPN. So also both for the ISPN, the contralateral choice activity is higher than the ipsilateral choice. But if we look at the contra preference, you can find the difference between the contra preferring DSPN and the contra preferring ISPN, and also for the ipsi preferring neuron, of course the ipsi activity is higher. But if you calculate the ipsi preference for DSPN and ISPN, you can also find the difference for this. And also, we are really interested in the before choice period. So we further look at the before answer period, whether there's a difference for their choice preference. And for this one, the contra-preferring neuron, the DSPN is higher than ISPN. And for the ipsi-preferring neuron, it is similar that the choice preference was higher for the DSPN. So one thing about calcium signal is that they have relatively slow time dynamics, and so we further do the convol of the calcium to get the inferred spike rate, and use the inferred spike rate to calculate their before answer activity, and the result is consistent with the calcium signal result. So till now we found that despite the fraction of choice preferred neuron was similar for DSPN and ISPN, the activity level was different. And another possibility for one type of neuron has higher influence for the downstream area is that if the neuron fires together, they would likely to have more impact for the downstream area. So next we look at their synchronized level for DSPN and ISPNs. So for this plot, we calculate the co-activity level with the Pearson correlation for pair of neurons with different interneuron difference, interneuron distance. So you can see if the neuron is closer, their synchronized level is higher, and this is true for both DSPN and ISPN. And for all the recording session, and we can see a similar trend that closer neuron would have higher synchronized level. But the difference between DI spin and I spin can still tell from this plot, and you can see overall, despite the distance dependence, the synchronized level for DI spin is higher for the I spin. And this is for all the neurons. And we further look at the choice peripheral neuron. It is consistent that for both contra-peripheral neuron and for the ipsi-peripheral neuron, their synchronized level is higher for the DSPN than ISPN. So we found another difference between these two types of neuron. And next, we want to have a global view. So we do the population analysis for the DSPN and ISPN. We build a pseudo population from the recording result and use the DMX PCA to derive the choice subspace. And here we show the first three PCA of the choice subspace, for example, DSPN population. So here the red line shows the trajectory for the contralateral choice, and the blue line is for the ipsilateral choice. And the circle indicates their answer time. You can see before the answer execution, these two trajectory was already separated. And this is true for both DSPN and ISPN. And for quantification, we calculate their trajectory selectivity for their for the contralateral choice versus the ipsilateral choice. And you can see from here the green line is for DSPN and the orange line is for ISPN. Compared with the shuffled, which we shuffle the trial label for the choice, and the DSPN, both DSPN and ISPN, their population trajectory selected for choice gets higher before the answer, but there's still difference for the DSPN and ISPN, and also this is true for the before answer period. So to summarize our result, for the optogenetics result, we found that this opposing effect of DSPN and ISPN is also true for the perceptual decision making. And also the causal effect for the ISPN is more like a decision epoch specific. And also the inactivation result shows that the shift of category boundary indicates it is more likely they involve in the decision formation process. So overall, our result reveal this opposing and coordinated contribution of the ISPN and ISPN, specifically in the perceptual decision process. And here, from our result, we also propose a conceptual model that both the ISPN and ISPN, they comprise multiple choice coding example. The red is contralateral and the blue is ipsilateral preferring. So from the opposing role of these two pathways, this ensemble forms a multi-ensemble balance, but the balance was not last forever. To make a decision, this SPN ensemble display a dynamic shift in their coding strength and also the coactivity to fairing to support their desired action. So I want to really thanks for my supervisor Nilong, and he is really supportive and considering. And also my collaborator, especially Cui Lele. So we share a equal contribution for this study, and also all the members of Lab and also my mice. And so at last, thank you for your listening, and happy Spring Festival. Yeah, that's all.